Note taker 11 begins our genetics unit. Genetics has to do with the traits, your characteristics that are passed down from your parents to you, and they make you the way you are today. We're going to start off our genetics unit talking about the structure of DNA. DNA stands for deoxyribonucleic acid. This is a chemical makeup that is found in your chromosomes, inside of the nucleus, inside of your cells. This DNA is what stores and passes on your genetic information from one generation to the next. Your DNA was discovered actually many years ago by a young lady named Rosalind Franklin. Now, at the time, it wasn't usual for a woman to actually have a job. During this time of in our history, women were more apt to stay home, cook, clean, while the man goes out and works. Now, she used a process called X-ray crystallography, and she was able to discover the structure of DNA. Now, we also had something unfortunate happen to Miss Franklin. She died at the early age of 30 from cancer. But the work that she had done, there were two other scientists that used this work that she had created or had discovered, James Watson and Francis Crick, and they built upon her work, and they went on to win the Nobel Prize for their work on the structure of DNA. So not only did they show what the shape of DNA was, but they also showed that the chromosomes inside of your nucleus are made of DNA. The structure of DNA actually looks like a spiral staircase, and we call this a double helix. Helix means spiral, and double because it's got two sides to it. So it actually does truly look, it almost looks like you took a ladder and put a twist in it. And the ladder of DNA actually is made up of many chemicals, and the bases, which make the rungs of the ladder, are made from 4 chemicals: adenine, thymine, cytosine, and guanine. Now, these chemicals are like puzzle pieces. They only fit together a certain way. So adenine can only pair up with thymine, and cytosine can only pair up with guanine. We're going to do some work in class that shows this. We use capital letters to designate each base: A for adenine, T for thymine, C for cytosine, and G for guanine. On this slide, we have a drawing of what the double helix shape of DNA looks like. And you'll notice that the rungs of the ladder, so if you look across, there are 2 colors per rung. You'll notice that some of the rungs are yellow and green, while others are orange and pink. You will notice that you won't ever see the yellow with the pink or the orange. You won't ever see the green with the pink or the orange. And you also won't see the colors with themselves, so you'll never have a yellow and a yellow together because 2 adenines can't go together. So this is how DNA fits together, and each of those is just a chemical bond there in the center. In 8th grade, you'll be learning more about chemical bonds and how they hold together. But with this, you'll notice that there's also an order to the rungs. So you might have a pink and orange, then you might have a yellow-green, you might have another yellow-green, or you might have an orange and a pink. So that pattern that you're going to form on the DNA is going to be something we're going to talk about later in the notes. Well, it's later in the notes, so we're going to talk about it here. Along with discovering or winning the Nobel Prize for determining the shape of DNA, also that your chromosomes are made of DNA, what Watson and Crick were able to show was that order, the yellow-green, or yellow-green, pink-orange, orange-pink, that pattern that those chemicals fell on the strand of DNA makes up a special code called our genetic code. And that is what determines what you look like, whether or not you have brown hair, black hair, blonde hair, whether you have blue eyes, green eyes, brown eyes, freckles, no freckles. It's a special code that tells our body what to look like. Now, you're going to have to think back to the mitosis note taker here. And one of the things we talked about on the mitosis note taker was in the stage of interphase. In interphase, the cell is resting, but the DNA is replicating, which means make a copy of itself. So we're going to talk a little bit here about how that DNA makes that copy of itself, or replicates. The latter is going to unwind, so the strand of DNA is going to unwind, and then it's going to be just like the zipper on your coat. A little portion of it is going to unzip. So the nitrogen bases are going to separate, so the adenine and the thymine are going to pull apart from each other. Now, once they pull apart from each other, you have all these extras, A's and T's and C's and G's, floating around inside the nucleus. These are called free nitrogen bases. And what's going to happen is they're going to float into the area that's just unzipped, and they're going to attach to the nitrogen bases that just unzipped. So where there's an adenine, which just unzipped from a thymine, a free little floating T, your thymine, is going to come in, and it's going to take its place and attach to that adenine. So once you have it all attached in that entire strand, it'll keep doing that all the way up the strand until the entire strand has replicated. Then you're going to end up with two new strands of DNA, which are identical to each other. Now, this is important in mitosis because if you remember, mitosis is creating new body cells for us. So my skin cells, I'm starting with one cell that has 46 of my chromosomes in it. And I need to form a second cell also with 46 chromosomes. So for this to happen, my DNA has to be identical when it makes a copy of itself. Now, here's a hint for you on how to remember which bases match up. A always goes with T, C always goes with G. So if you can remember where girls buy their makeup at CoverGirl, the A and the T go together, and the C and G for CoverGirl always go together. So that's just a little hint to help you remember what connects to which chemical. Here's a picture of DNA replicating itself. In class, we're going to be looking at a little activity on the smartboard where you're going to be able to drag over the bases to try to attach them and make new strands of DNA. Now, leaving our discussion of the structure of DNA, we're going to talk a little bit about the history of genetics. Now, we've already talked about how Rosalind Franklin did X-ray crystallography to find the shape of DNA, and how Watson and Crick built on her work and came up with their own ideas about DNA. But before we even knew DNA existed, we knew there was genetics. And we're going to talk long ago to the late 1860s, where we have a scientist. Actually, he's not even a true scientist. He was a monk, which is a religious man, but he also studied the plants and animals that lived in the monastery there. So we kind of call him one of the first biologists. Now, his name was Gregor Mendel. And by working with these plants in the garden, particularly the pea plants, because they're very fast growing and he could have lots of generations in one spring and summer, he was able to see patterns in the way characteristics, the flower color, the height of the plant, the way the flowers and leaves are arranged, the shape of the pea, the colors of the pea pods and the peas themselves, and he noticed there was a pattern. Now, we call these characteristic traits. You also have traits. Your hair color is a trait. Your eye color is a trait, and we're going to be talking more about ours later on. Now, what he noticed was when he noticed this pattern of the way these characteristics kept showing up, from one generation to the next, he said that these characteristics must be passed down from the parent plants. Now, all his work that he did with genetics, he actually became known as the father of genetics. Now, as he studied the certain traits, like the shape of the seed, which is the pea that we would eat, that's the seed of the pea plant, the seed color, the pod shape, how tall the stem was, as he bred more and more generations of pea plants, he noticed that some traits kept on showing up, while others would disappear for a few generations and pop up later on. So he came to the conclusion that every trait that we have, it has to have two factors for each of those traits. One of those factors comes from the mom, one of those factors comes from the dad. And he called these factors genes. And genes are units of heredity. Now, when we have a mom plant and the dad plant, the male part of the flower and the female part of the flower, each one of those is going to send down half of their chromosomes. So when we talked about meiosis, where you had a regular body cell having 46, but your sex cells have 23 chromosomes, that's because each of the parents' pairs had to separate. So when mom has 46 chromosomes, they all hang out in pairs. Well, mom can only send you one from each of those pairs. Dad can only send you one from each of his pairs. So mom's 23 plus dad's 23 make your 46. Now, with these genes, if you have true breeding plants, which means they have opposite traits, but one has both tall genes and one has all short genes, the baby plants that were made, once they grew up, all of the offsprings showed only one of the traits. So he concluded that one trait must be stronger than the other. The stronger trait is called the dominant trait, and that's the one that will always show through. The weaker trait is called your recessive trait. Sometimes that recessive trait shows, but other times it seems to disappear and doesn't show up for generations later. So for an example, freckles. Having freckles is a dominant gene. So if you have freckles, it means that one of your two genes has to be a dominant trait for freckles. If you do not have freckles, that means you have both recessive traits that code for no freckles in you. Here are some of the traits that Mendel observed in the plants. And you'll notice that for each trait or characteristic, like seed shape, seed color, you'll notice that each one is followed by a dominant trait and a recessive trait. So if we look at seed shape, having a perfectly round pea seed, that it's called spherical, which means perfectly round like a ball, that's the dominant trait. But sometimes you would have a wrinkled looking pea seed, that's a recessive trait. Seed color, look at what the dominant seed color is. The dominant color for peas is actually yellow. But you don't see that very often in the store buying yellow peas because green is a much more pleasing color for us to see when we're eating them. Flower color, purple is dominant, white is recessive. Now, as we look at these, since each trait each characteristic has two genes that code for it, we can have three possible different patterns for what your gene pair can be. So looking here on this slide, we have a gene pair for each of our characteristics. Now, we are going to represent dominant traits by using capital letters of the alphabet. We're going to represent recessive traits by using lowercase letters of the alphabet. Now, if you're talking about the same characteristic, just the two opposites in it, you do have to use the same letter of the alphabet. So the example I put here in the notes, if we're talking about the stem length in plants. Tall plants is dominant, so we give that a capital T. Shortness in these plants is a recessive, so we're going to use the lowercase of a T because we're still talking about the height of the plant, so we need to use that same letter. Later on, you're going to see in later notes as we continue with our genetic units, there are situations where we will have two different letters in a pair, but for right now, what we're going to be working with are the same letters, just one capital, one lowercase. So, I said that you have a gene pair for each of your characteristics. If you have two dominant traits represented by two capital letters, or if you have two recessive traits represented by two lowercase letters, you are what is called purebred or homozygous for that trait. It means that the two genes you have in your gene pair are the exact same thing, both dominant or both recessive. And when you go back to class, if you look on the word wall, the prefix homo means the same. So when we have a homozygous gene pair, we have two that are the same case and the same letter. If you have a gene pair that has one dominant and one recessive together, so that would be one capital and one lowercase of the same letter, then you are what is called hybrid or heterozygous for that trait. And again, if you refer to the word wall in the class, hetero means different. So if you have a capital T and a lowercase t, that's different from each other. One is dominant, one's recessive. And I mentioned that we'll also see where you can have two different letters in a pair, but that's going to come later on. If you have two different letters, even if they're both capital, if they're different in any way, you will have a heterozygous pair. Now, each of these different types of genes, dominant and recessive, when you have them that represent the same trait, like freckles, I can have dominant for freckles or recessive for no freckles. These are called alleles. So you can have a dominant allele or a recessive allele that will join together and make the pair. Now, as we continue on with genetics, we are going to do some more work with homozygous and heterozygous gene pairs. You are going to have to know the difference between the two because these two words we'll be using a lot in the future on our class activities. Please don't forget to do the 3-question assessment that follows this video.